All right, excellent. It looks like we are officially live and broadcasting to the YouTube universe. This is very exciting. Hi everyone and welcome in. I know it's about a minute after 11, but let's just give everyone maybe about 30 seconds or a minute or so just to get settled into this live stream and get excited for this really fantastic conversation that is coming up. Okay. Hi. It's so great to see all of you trickling into this live stream. Even though I can't see you, I know that you're there, and thank you so much for joining us this morning. Okay, excellent. So as you all are joining, let's go ahead and get officially started. We really don't have any time to waste because, like I said, we have a fantastic program lined up for you all. But I want to officially welcome all of you watching from home to another Labside Chat with the Seymour Marine Discovery Center. My name is Jen Cormier. And today's program is a really great opportunity to learn directly from a scientist with ties to UC Santa Cruz, and to really explore how scientific research plays a role in the understanding and conservation of the world's oceans. And before we really get to the meat of the conversation, I do have a really quick disclaimer right up top. This morning, we were experiencing some internet connectivity problems. So hopefully we don't run into too many issues this morning, but I do want to apologize in advance if any of you are experiencing any slow speeds or any audio issues. But if you do have any major problems, go ahead and add it to the live comments, and hopefully we'll be able to fix them. But I do hope that you stick around with us, because despite that, we have such a wonderful conversation lined up. We will be talking with and asking questions to Dr. John Carlos Garza, who I'm so excited to pick his brain and learn more about the research that he's up to. And so with that, before we get started, I also want to send a really, really big thank you right up at the top to everyone who actually submitted questions in advance for Carlos. You all submitted fantastic questions. There's so many, and I hope we have time to get through them all. But for those of you that didn't submit a question in advance for Carlos, but still want to pick his brain a little bit, feel free to add additional questions to the live chat as well, and we'll be able to answer those too. So let's just get on to it. So Dr. John Carlos Garza is a supervisory research geneticist, an adjunct professor in ocean sciences at UC Santa Cruz, and we'll be talking about various conservation tactics, life history traits, genetics research, fish populations, and the like. So Carlos, thank you so much for being here today.
It's really great to be here with you in this new virtual universe that we're living in. And one of the very few fun things about our new virtual lives is that we can choose where we want to be with our virtual backgrounds. And some days I'm in the Presidio with the Golden Gate Bridge behind me, and other days I'm in Australia. And so I'm pretending I'm in Australia today, which is actually a wonderful place for marine research, because in the waters, in the picture behind me lays the Great Barrier Reef, which is one of the world's most biodiverse sites, and really an amazing testament to the incredible biodiversity that exists in the oceans. And in spite of the fact that we here in the Monterey Bay area don't live in a tropical environment, we also live in a very biodiverse place as well, both terrestrially and obviously in the ocean. One of the more biodiverse, temperate marine ecosystems in the world is right here in Monterey Bay. So,
yeah, definitely, and we are just so lucky to reside on that little piece of land and see the incredible biodiversity that's just right off of our shores. So with that, so Carlos, I have a question for you. And so I said earlier that you are a supervisory research geneticist. So what type of questions are you trying to understand or answer with your research?
Great question. So the research in my group is actually pretty broad. We have a very incredible group of scientists and students from UCSC and NOAA that work in the lab, which is there on the Marine Sciences campus, as I'm sure you know. And the NOAA is the National Oceanic and Atmospheric Administration, which is the the federal agency that does most of the nation's marine biology and fisheries assessment and a lot of other things, weather included. Um, and so the unifying theme in our research is really the use of molecular population genetics, genetic data and analyses to understand populations of marine organisms, primarily fishes, but also some other organisms as well, including some of our local marine mammals and other things like cephalopods, but primarily fishes. And what I mean by that is that my group has a molecular biology laboratory with the ability to extract DNA from small pieces of tissue, isolate specific gene fragments or not, or just look at the entire genome, and then big DNA sequencers that sequence millions of DNA fragments in one fell swoop and provide us just with an incredible amount of data to understand the individuals, populations, species, and ecosystems that we study. So the other so people refer to us as geneticists, and that's because our toolbox is well stocked with the tools of modern molecular genetics, which is something that I latched onto early in my career, and actually early in the history of molecular genetics, believe it or not, when it just really at the dawn of it becoming possible to analyze DNA in an individual laboratory back in the late 1980s. And we have really expanded our research using that toolbox to study all kinds of different things, from the way that marine protected areas in the Monterey Bay are functioning to understanding what influences migration patterns in salmon and trout. And in fact, one of the things that is a kind of a unifying feature of a lot of the questions that we ask is migration, particularly of fishes, but how do they migrate? Why do they migrate? Why do they migrate? When they migrate? Where do they go? So on and so forth. And we study that on many levels, from just documenting patterns of migration by using what we would typically call in humans DNA fingerprinting to identify individuals at different times in their life, so we know where they have been and where they're going, to being able to then use the type of pedigree analysis that you have learned about in like CSI, that determines who the brother of the victim was or something like that, using DNA. And we can do that and understand not just where individuals move during their lives, but where their family members move. And so that allows us to follow families through multiple generations to understand how the patterns of migration are changing over time as well. And then more recently, with the genomics revolution and the ability to sequence whole genomes, we are looking at whole genomes and trying to understand the very fundamental biochemical basis of migration, finding genes and specific parts of genes that influence the timing of migration, the age at which fish migrate, and whether they migrate at all. For example, in trout, which probably many of you know, have the option to either move from their freshwater homes to the ocean and back or not. And whereas their cousins, the salmon, typically always go from the freshwater streams where they're born out into the ocean before returning to reproduce and die.
Wow, that is so incredible that you can learn all of this life history information from what I really like the term that you use, the DNA fingerprint, or kind of like, or fish CSI. So that's really, really incredible.
Finprinting.
And so when you're looking, yes. So when you're looking at these species and really trying to learn more about their migration, do you focus on a specific species, or are you looking at a broad spectrum? Excuse me. I know that you said that you're looking at trout, but is that specific, or are you looking at other species as well?
Well, we work on a number of different species. Our primary study species are salmon and trout, rockfishes, herring to a certain extent. Those are the main ones. And that's more than enough. There are many species, 3 or 4 species of salmon in California, and multiple distinct populations of salmon, and a similar number of trout species, and even more distinct populations of trout. And rockfishes have the center of their global diversity here in Central California. There are 50 to 60 species of rockfishes that live in the Monterey Bay region and the California Current large marine ecosystem. So a hyper diverse group of fishes, and they range from sort of large, somewhat solitary, bottom dwelling rockfishes like cowcod to schooling pelagic rockfishes like the short belly rockfish. So just a tremendous amount of diversity in that group as well.
Yeah, that's incredible. And, like you said, that's plenty. It sounds like you and your team have your hands full working with all of these different species. So that's incredible. So Carlos, I was reading an article about you the other day, and the article called you the fish matchmaker.
Oh, yeah.
And so is that true? And if so, what does that mean?
Well, so yeah, so a number of years ago, actually almost 20 years ago, we started getting involved with these efforts to rescue dwindling populations of salmon, endangered salmon, that were on the verge of being lost from some of the largest rivers in California, including the Russian River and the smaller rivers here in Santa Cruz County. And we noticed that, in fact, the techniques that were being used to rescue these populations often involved the captive breeding of these salmon, and that still occurs today in a number of places across the Western United States. And it occurs with a lot of other species as well, including the California condor and black-footed ferrets and a number of endangered species. And one of the things that became clear to me early on from my training in population biology is that that all species of animals that we have studied so far have mechanisms for identifying close relatives so that they can avoid mating with them, because, as I'm sure most of you know, mating with your close relatives is known as inbreeding, and inbreeding has a very predictable and severe detrimental effects on unmasking rare genetic diseases and a whole variety of other negative consequences. So, and when these captive breeding programs were mating fish, they were completely ignoring the fact that, particularly in these small dwindling populations, a lot of these individuals were closely related and even full siblings. So we devised, and it turns out, in retrospect, we realized this was the first time that this had been done when we did it back in about 2002, 2003. We devised a method to use molecular genetic data, DNA fingerprints, if you will, to identify the closely related individuals in these captive breeding programs and direct the individuals running these programs to not mate them together. So, and that sort of evolved into a whole scheme where every year we would collect genetic data for all of the individuals that were to be bred in those programs, and provide the managers with lists of the optimal mates, the least related individuals, so that they could not only avoid inbreeding, but also maximize the retention of genetic variation and of different types of crosses of individuals that will provide more variety in the population, and therefore more opportunities to successfully respond to environmental challenges. And so that really became something that is still going on today, unfortunately, because those populations are still endangered. But it's we've seen really big improvements in the overall survival and reproductive capacity of fishes in that with that breeding scheme. And in fact, it's now quite widespread and a fairly common technique. And we started it here in Santa Cruz. So
that's incredible. So now I know why the article called you the fish matchmaker, because you have one fish and you know exactly which fish that it could reproduce with. That is incredible that all of that information came from you and your team and your lab just right here in Santa Cruz. That's incredible. And so, Carlos, have you always been interested in genetics research and working with fish populations? Like, were you the kid that was jumping around in streams or always playing in nature, or did the spark come later down the road in your career?
Good question. So the two-part answer. The first part of the answer is I always knew I wanted to be a biologist from the time I was a little kid. When I was the family lore is that when I was 4, my grandma asked me, if I wanted to be like a firefighter or an astronaut. I don't know exactly, but when I grew up, and I said no, I wanted to be a research scientist and I want to work on the brain. And I definitely kept the faith in that, and I was one of like the few people in college who never wavered. I never changed my major. I always knew exactly what I wanted to do. But actually, when I was growing up, my stepfather and my best friend and his father were all commercial fishermen, and I didn't really. I mean, it was work and it was dirty and smelly, and it wasn't that attractive to me. And I'm not an obsessive fisherman, and so when I went off to when I started grad school first in San Diego and then moved to Berkeley for my PhD, I was working entirely on mammals, and I worked on monkeys, and I worked on mice, and then just kind of by accident, I started working on seals, and I started working on endangered seals in the Mediterranean, and then elephant seals, including right here in our area, and just by virtue of working on mammals that live in the ocean, next thing you know, I was sort of a marine biologist. And the jump from marine mammals to fish was not hard. And the tools that I learned and developed while working with mammals, including working with non-human primates, were really just powerful tools, and it was really fun to bring them over into the fish world. And because I had been working in, with human geneticists and other geneticists who were really sort of the bleeding edge of genetics at that time, we were able to bring innovations to the fish and wildlife genetics community that were really important and changed the way that people do business. And I mean, I think probably we just got there a little bit earlier than they would have because of that. But things like automating the DNA extraction process was something that, as far as we know, we were the first ones to do outside of the biomedical community. So no, I never thought that I would become a fish biologist. I had friends that were fish biologists in grad school and as a postdoc, including who worked on trout and salmon. And I never thought that I would, but then I saw the ad for this job that I currently have in Science Magazine. And it was all the same stuff I was doing then, but with fishes, and it was an easy fit. And so, and I never looked back. So.
Wow, that's incredible. And I just love hearing that answer too, because you also learned all of the innovation that you and your team did, as well as bringing everything that you learned over right from the biomedical field into this wildlife fish management world. And that's just so impressive to me. Like, my mind is blown. So that's very cool. And so we only have a few more questions left for you, Carlos.
Okay.
We are quickly running out of time, but I have a couple of really great questions that I want to ask you from the community.
Sure.
And one of the questions that I got, which is, I think, personally one of my favorites, was, do you have any myths or any, false facts that are floating out in the world right now about salmon, rockfish, or genetics in general that you finally want to put to rest or debunk?
That's a great question. We actually have a big paper that's about to come out about how about categorization of fishes and salmon in particular. And one of the things that I've been working on for years and that I've been trying to impress upon people is that we as biologists, as humans, are very much typological thinkers. We like to put things in boxes, and we like to talk about the Russian River salmon, or the Sacramento River salmon, or the Eel River salmon, or Scott Creek trout, or Waddell Creek trout, or whatever. And through our study of migration, one of the things that we've learned is that those boxes are more like milk crates. They're porous, and they're and the thinking about fish from and similarly fish from Carmel Bay versus from Monterey Bay, rockfishes, for example, it is it's misleading and often leads us to incorrect assumptions. Connectivity, networks of populations that are held together through migration, through movement and subsequent settlement and reproduction of fishes, is the reality of the biological universe out there. And we can lead ourselves into non-optimal solutions by continuing to think of, in particular, fishes, because they're so mobile, and leading into thinking that things are more different than they really are. That sort of looks can be deceiving, and even if fish in one river seem to be I don't browner than fish in another river, or have longer fins or whatever, often the causes of that are not profound, and are things that are not even necessarily genetic. And it can be the case that very, very simple genetic changes that are in fact things that actually are found that are variable within populations in the same way that like blue eyes and brown eyes are variable in human populations, can lead to profoundly different fish that do things quite differently, and people might consider to be different when in fact they may not very well be. So that's been the big myth, I think, that we've been trying to um, put into context, which is, things are more similar than they are different.
Yeah, definitely. Wow. And I really love that analogy that you use, that the box is like a milk crate where things are leaking out of it and whatnot. So that's a really great analogy to use. So my last question that I have for you, Carlos, time has flown by
Yep.
so quickly. But and I ask this to every scientist that I meet, but what is the absolute best part of your job? The reason why you love being a fish biologist.
Right. Well, that's easy, actually. I mean, we, and not very often, but we learn things about fishes and about the natural world using our tools that no one has known before. And the thrill of discovery, in particular, because of we're in a revolutionary period of being able to look at the very sort of foundation of life by being able to sequence whole genomes of individuals. And the thrill of discovery, of learning things about how things work in ways that, we're pretty sure that people have never, and no one has ever been able to before, is exhilarating beyond belief. I mean, it's really a fantastic feeling, and it's, I think, the thing that motivates most scientists. As I often tell my students, as they're trudging through another iterative data analysis or more experiments at the bench, we say, if you're lucky, it's 90% drudgery and 10% eureka, but that 10% eureka makes it all worth it. And I say if you're lucky, because sometimes it's only 2% eureka and 98% just hard work. But the eureka moments make it all worth it, for sure.
Oh, definitely. I mean, I can only imagine. And that's also really great advice to all of those aspiring young scientists out there too, is that, the hard work is worth it. And the sense of discovery is, it's mind blowing. So just keep pushing through. Well, Carlos, thank you so much for letting us learn more about your research and what you do, and even the incredible conservation that you are currently engaged in with being a fish matchmaker. So thank you so much. And I have hundreds of more questions for you, but unfortunately we are out of time. So do you have a place online or a website where folks can go to learn more about you and your research?
So for better or for worse, just in the last month or so during the pandemic, there's been a massive overhaul of all of our web presence, and we have not yet done the update of our website. So there's not much. Our old website is gone, unfortunately, that had a lot more information, including all of our publications. And we have not yet launched our new one. You can Google my name, or just go to the Southwest Fisheries Science Center website. That's probably the easiest. And find me, and then you can search for all of our publications and learn a little bit, sort of cursory, probably less than what I told you today, about our research, and also find out how to contact me. And if members of the public or the scientific community have specific questions or want to chat about things, then you can reach me by email, because I'm not in my office anymore, so you can't reach me by phone. Yeah, at least until the pandemic restrictions are over.
Yeah, definitely. And I will put those links in the video description below after we wrap up here too, so folks can easily find them. But yeah, that's really wonderful. So thank you so much for sharing all of that information. And before we go, I want to send a big thank you to everyone watching at home who tuned in today. It's really supporters like you that make these online programs possible. And so if you are in the position, I ask that you either make a donation to the Seymour Center or become a member with us so we can keep producing these online programs. You can find more information about donations and memberships on our website, which is seymourcenter.ucsc.edu. It's also, the links are also in the video description below, so take a look at that. You can also keep supporting us by subscribing to our newsletter to keep in touch with us, to learn about all upcoming events and activities. Keep telling your friends about us, sharing the word. We love to reach as many people as possible. So with that, I just want to say thank you again. We will see you next time for our next Labside Chat on October 22nd. So mark your calendars for that. But we hope you are all staying safe, staying healthy, and thank you so much again. Bye, everyone. Have a great day.
Bye.